So here we are with this year's Nobel. So here we are with our this year's Nobel Prize in Physiology or Medicine in 2025, right? So every year we don't give away these Nobel Prizes, right? But we know that there is a declaration and what you call as Nobel Prizes, they are given off, and this year's Nobel Prize of Physiology or Medicine, probably the field in which we interested, that is there in physics, it's there in chemistry, it's there in economics, and like much more, but literature and peace and all that stuff. But we are interested in the Nobel Prize of Physiology or Medicine of 2025. This year was given to Mary Brunkow, Fred Ramsdell, and Shimon Sakaguchi. All three of them, they are awarded this year's Nobel Prize for working on the phenomenon of peripheral immune tolerance, right? So a completely new area where actually these have opened up a space where we can now look at autoimmune disease in a different way. We can look at cancers in a different way. There are many things which have come up. Now we can actually deal and fight with many deadly cancers. We can fight with many autoimmune disease, before which this was a bit difficult. Now many what you call as drugs are now under clinical trials because of the discovery of this peripheral immune tolerance, right? So I'm not going to go into detail of all this, but to give a very brief overview, like what exactly happened, you can stay tuned with us, and we'll be coming with a more detailed view of how exactly this happened, right? So it was like, first I suppose it was Shimon Sakaguchi from Osaka, Japan, and he for the first time, he went against the tide in around 1995, right? We all know that we have an immune system which is protecting us from pathogens, and is essential. Like if a pathogen attacks and we are doing nothing, obviously we'll suffer from a disease, right? Pathogens need to be identified, and the crux is that these pathogens, when they enter our body, the fight is going on in our body. So the toughest challenge which our body has is to identify the pathogen and kill it selectively and does not harm our own cells, because you know that if our body starts harming your own cells, it will result in a serious complication of autoimmunity, right? So like it was known earlier also people knew that there is a what you call as mechanism of central tolerance, where when the immune cells are being made and matured in the thymus, that time there are certain cells which could be acting against our own cells, and these are potentially selected and they are destroyed so that you don't have cells which are harming your own cells, right? Because microbes are not coming in any uniform, so that you can identify, okay, this is microorganisms. They come in variety. Many of them are now actually mimicking our own cells. So our immune system is in great challenge to identify the pathogen and kill that pathogen specifically, right? So that is what is expected. And people found out that there is this central immune tolerance which is happening in thymus. But then it was Shimon who knew that immune system was much more complex than the central tolerance. He and his colleagues, and there was a class of people who actually were identifying certain cells which are actually what you call as killing those cells which are harming our own cells. But many people were not convinced that there are certain such cells which are actually present. But it was Shimon who identified that apart from this central tolerance, there is an unknown class. He by that time had not identified which is that class, but he told that in 1995 there is a more complex mechanism to this, and he came up with the idea of protecting our cells with this unknown class of T cells, but regulator T cells, which we now call. But that time it was not very clear. But then he was sure that there is a class and that is working as a peripheral immune tolerance. So it was this Shimon who came up with this idea that there is an unknown class of these T cells. In around 2001, almost 6 years later, we had these two other laureates, Mary Brunkow and Fred Ramsdell, working somewhere else, and they found mice who was like a strain of mice which was more what you call as prone to autoimmune disease. As I told you, I'm giving you only just idea like how it happened. We'll be discussing more about this in detail, like how the things went. Up till now, we came to know that Shimon knew that there is a special class of cells which is protecting us from killing our own cells, right? It was this Mary and Fred who came up with the idea that there is a strain of mice which is more vulnerable to autoimmune disease. It had that flaky skin and all. We'll discuss in more coming videos. You know? So they found that this specific strain of mouse, it was more vulnerable to autoimmune diseases. And they found that there was a gene called as, which later on identified as FOXP3 gene. And there was a mutation in this gene. The FOXP3 gene got mutated and the mouse was now developing an autoimmune disease. So you're getting my point. Two things happening. Shimon identified that there is a special class of cells which is protecting us from autoimmune disease. Mary and Fred, they identified a mice which is suffering from an autoimmune disease because of mutation in a particular gene. 2 years later, it was this particular person, again Shimon, who actually could relate these discoveries. He found that the FOXP3 gene, in which there was mutation, which was found out by this Mary and Fred, was the same gene which was coding for the same cells which were hypothesized by himself. He had told, no, there are certain cells which are protecting us. And this FOXP3 gene was actually the gene which was coding for your immune tolerance, like for T cells, for regulatory T cells, which are actually involved in protecting you from autoimmune disease. So now this mice has a mutation in this FOXP3 gene. And once there is a mutation in this FOXP3 gene, you'll find that the mice is now more vulnerable to autoimmune disease. That means FOXP3 gene now cannot produce a regulatory T cell. And this regulatory T cell was actually protecting from autoimmune disease. Right. So therefore, now we can have now more what you call as trials about working on this regulatory T cells, coming up with the medicines, you know, which could treat the autoimmune disease, which could treat the cancer. So what was not known up till now was all linked up by the discovery of Mary and Shimon. So as I told, like if this got messed up, we'll be discussing more in detail how exactly it happened. Right. So this was this year's Nobel Prize 2025, and stay tuned for a more detailed description of exactly how they came up with these ideas. To sum up in 1 minute, I'll just summarize what happened. Shimon identified a cell which is a regulatory T cell which is protecting us from an autoimmune disease. Mary and Fred, they identified a mice which had a mutation in FOXP3 gene, and mutation in FOXP3 gene was actually responsible for autoimmune disease. It was Shimon who again told that if this gene is mutated, the mice suffers from autoimmune disease. So it is the same gene which is coding for regulatory T cells. So regulatory T cells are playing a central role in protecting us from which diseases? Autoimmune. And now we can like handle transplants more easily. We can handle autoimmune diseases easily. We can handle cancer easily. And the world is now like having many drugs under clinical trial based on this particular hypothesis of these 3 Nobel laureates of 2025. Okay?